Hello everyone, welcome to today's live broadcast, Obtaining Non-Invasive Readouts in Early Safety Signals from Rodent Models. I am Christina Mahalik of Labroots and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by Labroots and sponsored by Byam. Byam is a Silicon Valley-based technology company applying digital technology into in vivo studies. They have developed a novel digital Vivarium platform incorporating HD video cameras and remote sensors for 24/7 recording of experiments, along with software and video processing algorithms to track and extract novel metrics and readouts. For more information, please visit Vium.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. Lastly, this is an educational web seminar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your webpage and follow the process of obtaining your credits. I would now like to introduce today's speaker, Dr. Laura Schaevitz. Laura Schaevitz has more than 15 years of experience in vivo animal studies. As VP of Scientific Technologies at Byam, she oversees the development of animal models and draws upon her experiences as a molecular and behavioral neuroscientist to direct the application of Byam's digital metrics to the animal models. Her past experiences include work across a broad spectrum of models, including transgenic and nutrition-induced models of neurodevelopmental disorders. I will now turn it over to Dr. Schaevitz for her presentation. Doctor, the floor is yours.
Thank you for joining me today. I'll begin by reviewing many of the challenges inherent with using animal models to predict efficacy and safety of compounds during the drug development process. I will then introduce an innovative technology platform that Byam has developed to address these many challenges throughout the continuous collection of the physiological data throughout the course of the study to gain insights. Finally, I'll demonstrate a few of the ways that Byam is using this platform to discover insights in specific disease indications. The prediction of efficacy and safety of potential therapeutics in preclinical rodent studies is challenging. The challenges can arise from both the applicability of animal models to the human condition, as well as in study design and execution. Obviously, rodents are not humans. As such, disease and injury models in rodents opt to capture one or two of the disease features, but they do not capture the full complexity of the disease course or its mechanisms. In the context of drug development, researchers must carefully pick models that have both known drug mechanisms of action that are likely to be translatable to the human, as well as mechanisms against which their drugs are predicted to act. There is an added complication that interspecies variability between pathways and biological systems need to be taken into account. As a few examples, interspecies variability in the immune system between rodents and humans complicates the assessment of immune-mediated diseases and the assessment of biologics in those animals. To ameliorate this problem, much work has gone into the development of mice with human immune systems, as well as in other humanized organ systems. Another example is in the assessment of drug side effects. CNS side effects account for attrition of drugs during clinical trials and upwards of 10% of the drugs which are withdrawn from sale. Here, Mead and colleagues took an unbiased approach to assess whether traditional rodent functional observational batteries, or FOBs, or Arwin tests could have been used to predict these adverse events using data collected from 5 pharma companies. The key takeaway messages here are that the rodent tests were able to catch serious side effects such as ataxia, seizures, which account for a high translational value. However, there was a class of side effects such as dizziness, nausea, headache, shown over here, that the traditional FOB and Arwin batteries weren't able to capture. This is perhaps not unsurprising given the interspecies differences in the presentation of nausea, for example. Rather, the authors suggest that perhaps a novel approach, such as through the continuous monitoring of rodents, may have been better able to detect these physiological changes that correlate with these adverse events. The second main challenge arises around study design and execution. In order to move the discovery of new drugs forward faster, you can think of the pharma companies, biotechs, and academics around the world as testing compounds in parallel, if you like. Obviously, reproducibility amongst all of these various companies is a main concern, and it's not just the in vivo studies, it's also the in vitro studies. The extent of this concern is evidenced by the large number of influential papers which are now coming out from key pharmaceutical leaders such as Bayer and Amgen, as well as in key initiatives by NIH, which cite that upwards of 50 to 90% of the published studies are irreproducible. Irreproducibility is often cited to be the result of many factors, including differences in study design, in execution and analysis, and this may result in different study outcomes depending upon the location for the same compound. Importantly, the call is never to limit science, but to improve the study design and to provide more data and the ability to record and monitor these parameters which may affect the study results. So one example of a lack of reproducibility was highlighted in this paper by Stephen Perrin and colleagues at the ALS Institute. Here they attempted to reproduce data from over 100 compounds that had been published as showing positive effects in ALS animal models. They were not able to reproduce the effects shown in the literature. In fact, many of the compounds actually showed no positive effect upon retesting. In looking at the studies, it was clear that a more careful design, identification of perhaps more translationally relevant outcome measures, and better powered studies may have improved the overall experimental results in those initial experiments. One of the study elements that researchers are unable to currently track well is the environment. So while it's easier to keep variables constant while testing compounds on rodents, in comparison, of course, to trying to track these variables in humans in clinical trials, there's still a large number of generally untracked factors that are predicted to impact the overall outcomes of a study. Many of these may be obvious, including the apparent smells, sounds, perhaps even lighting levels in the cage. As a behavioral neuroscientist myself, I can tell you I've heard many behavioralists who lament that depending upon rack position, whether it's the front or the back, or the top or the bottom, an animal may perform differently on a specific task. Disruptions to circadian rhythms due to entry of animal rooms after hours, the timing of dosing, all of these are known sources of variability. Standardizing all of these variables in an animal study across the world is certainly unrealistic, and one may predict that it would also lead to stifled discovery of new drugs. But what if a scientist could precisely track and account for all of these study variables during data analysis? And that's where the Byam digital platform is well suited. We started by building a better cage, drawing upon expertise in sensors and in the collection, storage, and analysis of large quantities of data. We created a system that monitors every aspect of an animal study continuously and automatically. The racks are designed to monitor precise environmental conditions at all cage positions. Gas sensor modules monitor temperature and humidity, while individual cage illumination provides uniform environmental conditions for all animals. All environmental condition data is piped through and stored with a study record, so if there are any abnormalities, you'll know about it. Video cameras are backed by computer vision algorithms that provide physiological data and are able to pull out meaning. We've developed an in-cage scale, which combined with conventional in-life measures and biomarkers to give you a comprehensive view on how a drug is affecting an animal. It's digital medicine for preclinical studies, and we collect vastly more information than has ever been possible before. We deploy these cages at scale. We have thousands of them operating today in Vium's digital vivarium in the South Bay. All the data from these experiments are shipped to the Byam cloud, where it is stored and analyzed. Studies are accessed through our online research suite, where a researcher can design, run, and analyze an experiment from a laptop anywhere in the world. Results are available in real time, even while the study is running. To give you an idea of what our online platform looks like, you can see a slide shot here. Data is available both for individual subjects on the subject view or across a full study in the analytics studio. On the subject view, you can explore a full video record of the entire study by selecting on regions of interest in the time selector here, or by exploring specific data points by clicking on areas of interest on motion here, breathing rate here, or other graphs not shown here, which include temperature, humidity, subject observations. In addition, in the analytics studio, here we see the analytics studio. You can explore data in easily downloadable graphs, complete with statistical tests, or explore the data on your own by downloading all study data, including hourly motion, breathing rate, and conventional studies, or conventional measures. We've developed a number of metrics that we use to monitor the animals continuously. Two of the measures shown here are relevant across almost all of the disease indications, and they're basic measures of motion and breathing rate. And both are derived from video using complex computer vision algorithms. So for example, to measure motion, the direction and velocity of motion between video frames is calculated at defined points in the video image. All of these small dots you see under computer vision analysis. The velocity is then used to create a frame-by-frame summary of motion, which is read out in the activity summary, with the highest speeds reading out as the brightest blue spots. Here you can see that the brightest blue spots correlate with the location of the mouse. We calculate the speed on every frame. In total, we actually acquire 86,400 data points per day per animal. That's a lot of data points. On the individual subject graphs, we present motion, which is averaged into small time windows on the order of minutes. But for analysis, we can analyze motion in any way imaginable, including averaged by day, night, or in any time window which is appropriate for a specific study. For more information, as well as additional validation data, please see Vium's website. To measure breathing rate, we use points of time when the animal is still for a greater than 30 seconds. After identifying the animal, you can see outlined in the orange box on the video frame, the strength of periodic movements that fall within rodent breathing rate frequency bands are then plotted as individual data points on the subject view. Shown on the far right is data from our validation study run in mice to demonstrate that the computer vision algorithms accurately measure breathing rate. Here we took C57 BL6J or C3H mice and placed them inside of a plethysmograph inside of our Vium cages. We then compared each computer vision generated breathing point to the breathing rate used, computer vision breathing point to the breathing rate using the plethysmograph, and we found that the rates were highly correlated with an R-square of 0.98. On average, we found that C57BL6J mice showed a significantly higher breathing rate at around 180 breaths per minute versus the 136 breaths per minute in C3H mice, which is consistent with the literature. Again, please feel free to visit our website for additional validation data and the relevant literature citations. I now want to take the opportunity to share case study data collected on the Byam platform from 2 of our animal models, including lupus and lung injury models. These models, while predictive, both have limitations that make running the model labor-intensive and yield few in-life measures against which to assess drug efficacy. Let's begin with lupus. Lupus is a heterogeneous disease which manifests in humans with a number of different clinical signs that include renal failure, hair loss and rashes, painful and swollen joints, all the way to CNS symptoms including anxiety, fatigue, and potentially cognitive dysfunction. Importantly, the number and extent of these clinical symptoms varies among individuals with lupus. So not all individuals with lupus have all of these symptoms. There are several animal models whose pathology mimic these many aspects of the human disease. Current conventional measures in these models focus on assessing a subset of the clinical symptoms in life, including renal failure as measured by proteinuria and autoantibody production, and then assess tissue histopathology at study end. While a few labs are beginning to explore the translational value of some of the more CNS-specific side effects, these studies are currently very labor-intensive and not easily used for assessing drugs at scale. In addition to the limitations in the ease of collecting biological insights into some of the more complex disease states, there is the additional downside that lupus models are very long to run. Standard spontaneous genetic models, such as the MRL, LPR, and NZBW models, can take upwards of 10 to 20 weeks minimum for the in-life phase, with additional time at study end to conduct histopathological assessment of the tissues. As an example, here today I'm unable to show you any histopathology because while the study was completed more than a month ago, we're still waiting. Given the length of the study, there is the added cost of maintaining the animals and the significant amount of compound required to dose the animals for over 10 weeks to assess efficacy. To give you an idea, in this study we used a conventional MRL-LPR model of lupus. This was a traditional prophylactic study in which the treatment of 2 reference compounds were begun prior to disease onset. To orient you to the study design, we ran the study in a very conventional way. Looking at the timeline, the animals were brought in-house at 6 weeks and were allowed to acclimate until 9 weeks of time, at which point we began to dose with cyclophosphamide 1 time every 7 days or with dexamethasone 3 times per week. We measured conventional endpoints throughout the study, including body weight and proteinuria weekly throughout the course of the study. At study endpoint, we collected spleen weights and also kidneys for histopathology. On top of the standard study, we overlaid the Vium platform. So prior to beginning dosing, we randomized animals into treatment groups using both motion as well as body weight. Then throughout the course of the study, we continuously collect motion and breathing rate data, as we were able to assess, in the few instances where animals were found dead, the circumstances of death by a retrospective analysis of the videos. In all of these cases, we found that the animals died following severe, unexplained convulsions. Here we demonstrate the disease incidence in the MRL-LPR mouse looks similar to the literature in our hands. The dotted lines on each of the graphs shown here represent the start of treatment. Vehicle-treated MRL-LPR mice, these are the gray lines, we found that body weight increased starting around 12 weeks of age compared to the control, which are the dark black lines. Both the reference compounds, dexamethasone in blue and cyclophosphamide in orange, show less severe body weight increases compared to the vehicle-treated MRL animals after 15 weeks of age. Proteinuria scores are also significantly elevated in vehicle-treated MRL mice after 4 months of age, and this is again significantly ameliorated by both dexamethasone and cyclosporin at 5 months of age. Looking at Byam metrics, they show a similar disease incidence and course. Again, on both graphs, we represent the time of treatment as started by the dotted line. Given that this is prophylactic dosing, I'll remind you that we do not expect animals to show signs of disease at this stage, and you can see prior to that dotted line relatively consistent motion and breathing rate data at that age. Focusing on the graph on the left, you can see that motion decreases over time for all groups, including the dark-lined controls. Decreases in motion over time is actually standard across many of the studies that we run at Byam and is associated with aging. However, you can see that the vehicle-treated mice, again, these are the 2 gray lines here, showed significantly lower levels of activity than in either the dex or the cyclosporin groups the blue and the orange groups beginning at 13 weeks of age. In fact, we found that motion in the cyclosporin and dexamethasone treated groups are not significantly different than control mice. Unlike motion, breathing rate actually increases over time in the vehicle-treated mice. Shown here, you can see that starting at around 14 weeks of age, the vehicle-treated animals have significantly elevated breathing rates. But again, cyclophosphamide and dexamethasone-treated animals, the blue and the orange lines, are no different from the genetic wild-type controls shown here. I now place the conventional measures of body weight and proteinuria next to Vium's motion and breathing rate metrics for your comparison. We find that the Byam metrics of motion and breathing rate track disease progression similarly to the conventional measures, with one very notable difference. We find that we begin to distinguish groups a few weeks earlier, specifically at 13 and 14 weeks of age, for the Byam metrics compared to the 15 to 17 weeks of age for the conventional measures of body weight and proteinuria. This data suggests further that the MRL-LPR model of lupus, that the Vium digital vivarium and Vium's metrics can be used to effectively distinguish therapeutic efficacy of several reference compounds. Vium's platform provides insights into physiologically relevant measures which track disease and reflect drug efficacy, such as the motion and breathing rate, both of which provide a more holistic overview of disease state in SLE. Increased breathing rate may be a reflection of pulmonary edema or acute reversible hypoxemia secondary to renal failure. It could be due to interstitial pneumonia or increased cell infiltrates in lungs, or it could be a general immune-mediated response, which has been reserved in lupus patients. Although its pathophysiological relevance to disease requires further investigation in our hands, breathing rate here does appear to be a sensitive metric against which we can assess drug efficacy. Now we move to the lung injury model, which presents many similar challenges to lupus models in the availability of easy-to-acquire in-life measures. A number of lung injury models have been developed to assess various mechanisms of lung injury based on reproducing known risk factors for acute respiratory distress in humans. A physiologically relevant measures in life or a more physiologically relevant measure in life is the actual measurement of lung function through highly labor-intensive analysis of respiratory function, as are sometimes measured on devices such as the plethysmograph. Or researchers may choose to focus on lung histopathology assessment at the end of the study. We, however, hypothesize that the continuous measurement of breathing rate may provide an easily accessible measure using the Byam platform. For the model we show you today, we use a paraquat-induced lung injury. Again, to orient you to the model, here paraquat administration leads to a loss in pulmonary function and inflammation that is coupled to the loss of mitochondrial DNA integrity and eventually to the permanent loss of lung tissue. To orient you to this study, we used Lewis rats between 300 and 350 grams. The rats were acclimated to our facility for 7 days, at which time they were administered a single intratracheal dose of paraquat or saline. We track conventional measures including body weight and body temperature both before and after lung injury. On top of the standard study, we again overlaid the Vium platform. We found the breathing rate in all rats was very consistent. Therefore, when we randomized animals, we based it off of motion and body weight. We then monitored motion and breathing rate both before and after a lung injury. Here we demonstrate again that conventional body weight and body temperature demonstrate a robust induction of lung injury. Body weight and body temperature is plotted as a function of day, with again the dotted lines shown here as the day of induction. Body weight was significantly, as you can see, body weight prior to induction is similar in both groups. But body weight significantly decreases 2 days post-injury in the paraquat-injected animals. Those are the blue lines compared to the controls in the black. But they begin to recover on days 5 and 6 post-injury. Body temperature is also significantly decreased on days 2 to 4 after injury, but recovers to normal by 5 to 6 days post-injury. Now we look at Byam metrics. Here, the breathing rate and motion are plotted in a slightly different way. They're plotted as 4-hour averages such that you can see circadian changes in activity and breathing rates. If you focus first on the motion graph to the right, you can see that nighttime is represented as the dark bands, where my arrow is, and that the time of lung injury is again marked as this faint dotted line shown here. You can see a clear pattern of day and nighttime activity representing the circadian rhythms prior to injury. About halfway through the first evening after injury, around 16 hours, here, there is a significant drop in activity in the paraquat rats, which persists for several days. This drop begins to recover, although not fully, until days 5 and 6 post-injury. The breathing rate shows a much more persistent effect. As early as 12 hours post-injury, breathing rate was significantly elevated by as much as 2 times the baseline rate. This effect persists throughout the 6 days that the animals were followed, with no amelioration. The effect here was actually so significant that power analysis using this data suggests that we would only have required 2 to 3 animals to achieve highly statistically significant results. Again, comparing the conventional body weight and body temperature measures to conventional measures, we note the following. First, that the pattern of motion changes tracks quite similarly with body weight and body temperature, providing additional support for conventional measures. And we find that motion and body weight are both more sensitive measures than body temperature, and that these measures may be a direct reflection of the inflammatory responses in these models. Second, the motion and breathing rate changes are present earlier than conventional measures. Here we find that significant decreases in body weight and body temperature are not evident until 2 days or more than 48 hours after lung injury. However, in contrast, we were able to detect changes in the biometrics as early as 12 to 16 hours after insult. And third, unlike other metrics, breathing rate, which is more physiologically relevant metric, shows this persistent elevated levels, which should more accurately reflect changes in lung function that result from paraquat injury. To sum up a few of the findings from the lupus and the lung injury studies, as well as studies that I didn't have the time to discuss today, we demonstrate how it's possible to use fewer animals when we use more physiologically relevant in-life measures of breathing rate, for example, in the lung injury model. Vium metrics often detect also differences between groups earlier in both cases here in the course of the study than the conventional measures. In addition, our continuous monitoring ensures that changes in environmental conditions that may change study outcomes can be tracked and accounted for during study analysis. There were no abnormalities in these studies to show you. As we run additional studies across a number of therapeutic areas, we find that many of the Byam metrics are very consistent, both in the time to onset as well as in the magnitude of change. We hope that this will allow us to begin pooling historic data to reduce overall control numbers required in future studies. And finally, by using predictive analytics, we have been able to anticipate when an animal is at the end of life so it can be humanely euthanized and properly studied before it's found dead in its cage. We do all of this while setting a new standard for animal welfare. Often there's a tension between science and animal welfare, but this technology allows us to increase this opportunity so that we generate wins for both the science and the animal welfare together. As a result, during our ALAC accreditation, we were given an unprecedented commendation for promoting the 3 R's. Returning to the challenges in preclinical studies, we're currently working on additional R&D projects to explore how continuous monitoring and biometrics can be used to improve the reproducibility of standard animal models and to provide insights above and beyond the conventional studies to improve the translational applicability of these models. In addition, we would like to begin to investigate how biometrics and insights can be used to create novel animal models or in-life measures that will further enhance the drug discovery process. To that end, we would love to work with you. If you're interested in working with Byam, there are 2 ways to engage with us and with the technology. First, we can run a study with you in our Vivarium. We've run a number of studies in the areas of autoimmune disorders, inflammation, organ damage, and phenotyping, as well as others. While we've developed a number of models in-house, one differentiator of our platform and our company is that we're excited to work with anyone to run standard or novel models on our platform. We would love to talk to you about any models that you feel would be well suited to the platform. To make the platform more available to anyone, we've also created a grant program open to both academic and private companies. Submit your proposals to develop a model on the Byam platform. We'll cover all the costs up to $100,000. You can visit our website to learn more. Thank you for your attention, and now I'll open up for any questions.
Thank you for that informative presentation, Dr. Schaevitz. Before we get started on the question and answer session, I'd like to remind our audience how to submit your questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as time allows. And with that, let's go ahead and ask our first question. The first question asks, what other types of models have you run on your platforms?
thank you for that question. We've actually run models across a wide variety. Other autoimmune disorders that we've looked at are MS. In the inflammatory space, we've looked at RA models, including both rat and mouse models of collagen-induced arthritis. We've also looked at IBD models, as well as a number of CNS models, including Niemann-Pick C disease. We've done a few experimental models in the area of glioblastoma as well. So we have looked at models in a fairly wide variety of spaces. And again, we're willing and interested in working in any new areas that you feel would be most interesting to you.
Thank you. Our next question asks, do you ever pair your system with telemetry devices?
Yes, in fact, we have. We do have a TSE system in-house, and actually during our early breathing rate validation studies, we did use both the plethysmograph and some of the TSE telemetry devices as means to compare our computer vision breathing rate metrics to these more gold standard methods. So we do have that capability, and if there are newer novel ways that you would like to use the telemetry devices, perhaps to look at heart rate or blood pressure or anything else, yes, we are more than capable and would be interested in working with you on that.
Excellent. Thank you so much for that answer. It looks like we only have time for one more question today. Our last question asks, how do I get access to your system?
That is a great question. We understand that scientists like to be able to explore new platforms, perhaps before they pay for them. Therefore, we've offered access to several of our internal studies upon request. You can go to vium.com and register to gain access to any of these studies. In those studies, you can explore and see all of the video and all of the data and all of the analytics that we've generated for those studies. So please feel free to reach out, and we'd love to give you access to some of these studies to explore.
Dr. Schaevitz, I'd like to thank you for your answers today, and additionally one more time for that great presentation. I'd also like to thank our sponsor, Byam, for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through May 2017. You will receive an email from Labroots alerting you when this webcast will be available for replay. We do invite you to forward that announcement to your colleagues who have missed today's live event. Until then, thank you for your time, and we'll see you next time here at Labroots. Goodbye.